Hello, bonjour. My name is Michelle Monje. I'm a associate professor of neurology, and I'm a pediatric neuro-oncologist at Stanford University. Today, I want to tell you a story in two parts about the way in which neurons interact with the glial cells that form the myelin sheath, how this contributes to central nervous system development and plasticity, and how these really powerful neuron-glial interactions are subverted in the context of childhood glial malignancies. But I like to start every talk with a broad consideration of neurological development. And I find the best way to do that is to show you pictures of my children. So this is my daughter, Sophie, when she was an infant. And we realized by the time we had Sophie that there is just so much neurodevelopment that human infants undergo every day in the first year of life that we had to make Sophie a onesie with a checklist of her neurodevelopmental to-dos. For example, human infants make something on the order of 500 billion new synapses every day of the first year of life, 14,000 new hippocampal neurons daily, 127 million new cerebellar granule cells. There's so much development in the posterior fossa during infancy. But perhaps the largest task that humans have in the first year or so of life is to myelinate their central nervous system. Humans are a semi-altricial species. We're born, with almost no myelin in the central nervous system. And then around the time of birth, myelination begins to occur in a massive wave over the first 2 years of life. It begins in the center of the brain around the central sulcus and then progresses towards the poles. It begins in the cervical spinal cord and then progresses towards the lumbosacral cord over that first year of life. And actually watching infant motor development over the first year of life, you can really see spinal cord myelination in real time, with humans first around a month or so being able to hold up their heads a little bit. And then as myelination progresses down the cervical spinal cord, by 3 months, being able to bring their hands together. By about 6 months, as that myelination continues down to the thoracic cord, being able to sit. And by about a year of life, being able to walk as myelination of the cord reaches the lumbosacral cord. But myelination does not stop after this first massive wave of development in the first 1 to 2 years of life, but rather progresses over almost 3 decades of human development, and in very predictable chronological and topographical patterns, such that circuitry that underlies more basic functions like sensation and motor function myelinates a little bit before those circuits that underlie more complex cognitive functions. So for example, there's a discrete wave of developmental myelination in mid-childhood in the brainstem, while during adolescence and young adulthood, the cortex and intercortical association fibers, so important for higher cognitive function, myelinate. And it's important to remember that this process of myelination, the process by which glial progenitor cells proliferate, generate new oligodendrocytes, and then form this insulating sheath around axons to decrease transverse capacitance and increase the speed of neural impulse conduction, it really does not end even at the end of neural development. In certain places of the nervous system, myelination continues throughout life, particularly in the neocortex. We see that there's this accumulation both of oligodendrocytes and of myelination. And in many regions of the nervous system, myelination remains somewhat incomplete, as though there's a degree of tunability to the system. And this is important because neurodevelopment is fascinating in its own right, but also critically important as we consider glial malignancies of childhood and adolescence. These occur in a very predictable spatiotemporal pattern that maps very well onto the spatiotemporal pattern of myelination, so that at a time when there is a discrete wave of myelination in the ventral pons, this is when one of the worst human cancers, diffuse intrinsic pontine glioma, tends to occur. And similarly, at a time when there is a discrete wave of cortical and intercortical fiber association in the cerebral hemispheres, this is when pediatric glioblastoma tends to occur. And these observations are concordant with discoveries from my laboratory and from many others that many forms of childhood gliomas arise from precursor cells in the oligodendroglial lineage, either oligodendrocyte precursor cells or OPCs, or just earlier precursors destined to differentiate along a glial lineage. And certainly fits with molecular observations about these gliomas, that there is a subpopulation of cells that at the single cell level very closely resemble normal oligodendrocyte precursors. And so we may glean really important clues about what regulates pediatric gliomagenesis by better understanding this process of gliogenesis. And this asks the very basic question: what does regulate the proliferation and functional differentiation of oligodendroglial lineage precursor cells? Well, one idea that's been in the literature for some time is the idea that neurons themselves may regulate the extent to which their axons are myelinated, and that this may occur in an activity and experience-dependent manner. This is an idea that until very recently was fairly controversial in the myelin field, in part because there are also quite clearly activity-independent modes of myelination. And so for the last decade or so, my laboratory has been studying the interactions between neurons and these oligodendroglial cells, asking questions about whether neuronal activity can modulate this process of developmental and plastic myelination, whether myelin may be plastic and adaptable to experience in adolescence and adulthood. And so I'd like to focus this brief first part of the talk on this idea of myelin plasticity, of activity-dependent adaptive changes in myelin that fine-tune circuit function. And to introduce you to the people who've led this work in my laboratory, this is Erin Gibson, who was my very first postdoctoral fellow. She is just a brilliant young scientist who's now leading her own research group at Stanford as an assistant professor. And this is Anna Gearty, who's still currently a postdoctoral fellow in my lab. And together, Erin, Anna, and several others have leveraged this really powerful technique called in vivo optogenetics, which allows for control of neuronal activity in neurons, in targeted populations of neurons expressing light-sensitive ion channels. And so here, if we express channel rhodopsin 2, this blue light-sensitive cation channel isolated from algae in cortical projection neurons, and then deliver light at the surface of the brain, we stimulate cortical projection neuronal activity in the motor planning area. And in a mouse in which there is channel rhodopsin 2 expressed in these cortical projection neurons, when we stimulate the motor planning area, at the frequency at which these neurons typically fire, the mouse executes complex motor behavior, which in a cage like this looks like walking in a circle. We have litter mate controls for these animals that do not express channel rhodopsin 2, in whom blue light delivery has no behavioral effect. And so when we see this complex motor output, we know that we've effectively recruited activity within this circuit, and then we can ask straightforward questions about how other cell types within the stimulated circuit respond to changes in neuronal activity. And what we found in doing this is that there is a rapid and robust proliferation of oligodendrocyte precursor cells, quite specifically within the stimulated circuit, and actually even more specifically within the corticocallosal limb of the stimulated circuit. These proliferating oligodendrocytes, if we track them over time, generate new oligodendrocytes, and that confers subtle changes to the myelin sheath. Changes that we would predict would alter circuit dynamics and influence function. And indeed, what we found was that these activity-dependent modulations in myelination within the premotor circuit improve motor functional performance, and that motor improvement depends upon the generation of new oligodendrocytes. And so these new oligodendrocytes either myelinate previously unmyelinated axons, previously unmyelinated regions of discontinuously myelinated axons, or remodel existing myelin. And these relatively subtle changes in myelination confer really important changes to circuit dynamics. They tune the circuit to promote coordinated circuit function. We've worked for many years to understand the molecular mechanisms mediating these neuron-glial interactions in the healthy brain. And one really key interaction that we discovered is activity-dependent BDNF, or brain-derived neurotrophin factor secretion from neurons, signaling to oligodendrocyte precursor cells. This is a required component of the activity-dependent mechanism in cortical projection neurons. There are also synapses between neurons and oligodendroglial precursor cells. Dwight Burgles discovered these over 20 years ago, and the role of these so-called neuron-glial synapses are still something that the field is trying very hard to determine. What we've discovered, though, while the mechanism of adaptive myelination is likely much more complex than just neurotrophin signaling, because this is a required component of the mechanism, that gives us a molecular handle to begin to ask what role myelin plasticity plays in normal brain function. And using that molecular handle, we've discovered roles for this adaptive myelin plasticity in various forms of cognition, including attention, memory, and learning. But the pressing question for pediatric oncology is what happens when these really powerful interactions between neurons and oligodendroglial precursor cells are subverted? What happens when these oligodendroglial precursor cells have an oncogenic mutation or have transformed into a glial cancer? So we'll focus the second part of the talk on this idea of malignant myelin plasticity, of activity-dependent growth of glial malignancies. And to introduce you to the people in the laboratory who have been leading this work, this is Hamsa Venkatesh, who was a cancer biology PhD student with me and then a postdoctoral fellow. She's now just left my lab to go begin her own research group at Harvard University. She's absolutely brilliant. And if there's anybody in the audience looking for a postdoctoral fellowship, I very strongly recommend Hamsa's new lab. This is Ewan Pan, who is presently a senior postdoctoral fellow in the lab, presently on the job market. And this is Tara Barron, who's a glial electrophysiologist who has recently joined our laboratory. And these are the diseases that we focus on, these malignancies of glial malignancies of childhood, which, like myelination, really occur in a very, very predictable spatiotemporal pattern, with low-grade gliomas of the optic nerve, for example, tending to happen in early childhood, midline gliomas, those characterized by the histone 3 K27M mutation, typically happening in the brainstem in mid-childhood, in the thalamus a little bit later in mid-childhood, and these hemispheric gliomas, which represent actually a collection of different kinds of molecularly defined diseases, occurring more commonly in adolescence and young adulthood. And so, we wondered whether these glial malignancies, many of which are thought to arise from oligodendroglial precursor cells, could neuronal activity similarly promote their proliferation and growth? And this idea that neurons and glioma cells may be interacting in important ways is something that was really, I think, suggested by the histopathology, something that neuropathologists have likely recognized for probably over 100 years, since the time of Scheer, when these secondary structures of Scheer were first described. This phenomenon of perineuronal satellotosis, the propensity of these malignant glioma cells to cluster in very tight microanatomical association around neurons in the tumor microenvironment, suggests that there's some important signaling relationships between these two cell types. And I want to point out that this particular H&E image comes from an early postmortem autopsy sample from a 5-year-old patient of mine back when I was a neuro-oncology fellow. And it was this child's tumor donation at the time of his death that allowed us to generate the very first cell culture and mouse model of diffuse intrinsic pontine glioma. And it's really through similar such tissue donations that we're able to do all of the work that we do. And so I want to just acknowledge that critically important contribution of patients and their families. And so to ask whether neuronal activity similarly drives glioma growth as it does normal glial precursor cell proliferation, we use that same optogenetic stimulation strategy, stimulating optogenetically cortical projection neurons in the motor in the motor planning cortex, or M2 cortex, but this time in the context of a diffusely infiltrating histone wild type cortical glioblastoma taken from an adolescent patient of mine. And what we found is that when we stimulate cortical projection neuronal activity, that just like their normal counterparts, the malignant glioma cells increase their proliferation rate, and this increases the overall tumor burden, specifically within the stimulated circuit. Turning to a different model, a low-grade glioma model associated with neurofibromatosis type 1, and in collaboration with David Gutman, who spent the last several decades developing very elegant genetically engineered mouse models of neurofibromatosis type 1. We find that if we modulate optic pathway activity, either optogenetically in this case or naturalistically, which I'll show you in a moment, if we stimulate the optic nerve using blue light delivered just behind the globe, specifically to these retinal ganglion axons, that has an effect of increasing the growth of the optic gliomas. Now, that optic glioma mouse model allows us to modulate optic pathway activity simply by manipulating environmental light. And so we find that if we take these optic pathway glioma models, these mice that in association with the NF1 mutant background develop optic pathway gliomas in almost all of the mice, very specifically around 9 weeks of age, if we place the mice in complete darkness, if we begin to dark rear them, just at the time that these tumors emerge or just after the time that these tumors emerge, we find that far fewer and much smaller tumors are generated in mice that are reared in darkness to decrease visual experience compared to their littermate controls raised with normal visual experience. And if we place these NF1 optic pathway glioma mice in the dark prior to tumor initiation, around 6 weeks of age, we find that no tumors form, despite the genetic predisposition for optic gliomas in this mouse model, suggesting that optic pathway activity is a critical regulator of tumor genesis in this context. So we wanted to understand the mechanisms that mediate these really powerful interactions between neurons and malignant glioma cells in the childhood central nervous system. To do this, we turned to a different experimental system to test the idea that there are secreted paracrine factors that may be quite important. We took explants of either healthy mouse cortex or healthy retina and optic nerves. And then in minimal media, in just artificial cerebrospinal fluid, we collected the secreted paracrine factors secreted into this conditioned medium when these explants were either spontaneously active, stimulated to increase activity optogenetically, or silenced using the voltage-gated sodium channel blocker tetrodotoxin. And we find that when we place this conditioned medium onto cultures of various forms of glioma cells, that there is a neuronal activity dependent increase in glioma cell proliferation that is lost when neuronal activity is silenced with tetrodotoxin. Now, many in this audience know very well that childhood gliomas are a really clinically and molecularly distinct group of diseases. But we find that this response to neuronal activity in general and to these paracrine secreted factors specifically is conserved across a wide range of different glioma subtypes, from histone wild type cortical glioblastoma to diffuse intrinsic pontine glioma and other midline gliomas, including also adult glioblastoma, anaplastic oligodendroglioma, and optic gliomas. Performing a secreted factor screen to try to understand what it was that was in this conditioned medium that was mediating this event, we find that there are two key glioma mitogens regulated by neuronal activity that we can discover in this experimental platform. One, not surprisingly, is brain-derived neurotrophic factor. Just as it influences the proliferation of normal glial precursor cells, it also very strongly influences the proliferation of malignant glioma cells. But really unexpectedly, we found this molecule Neuroligan 3 in a novel shed form. This is a postsynaptic adhesion molecule. And Neuroligan 3 emerged from the screen as the most robust glioma mitogen. Now, we know that Neuroligan 3, the synaptic adhesion molecule, is present at normal synapses, both those that are between neurons and other neurons, as well as those that are between neurons and glial cells. This is a well-studied molecule. It plays some role in some familial forms of autism, for example. It regulates synaptic strength, but it was not known to be a mitogen in any context. And in fact, it was not even known to be secreted. But we find that Neuroligan 3 is cleaved at the membrane, shedding this large N-terminal ectodomain in a strictly activity-dependent fashion through the enzymatic activity of the metalloprotease ADAM10. And so the next question is, which cell types in the tumor microenvironment are releasing Neuroligan 3? Well, we know that neurons express high levels of Neuroligan 3, and certainly neurons play a role as a source in secreted Neuroligan 3. But through cell type specific conditional genetic depletion experiments, we found that not only neurons, but really importantly, oligodendrocyte precursor cells, which again are a postsynaptic cell type, contribute to the secreted pool of Neuroligan 3 in the tumor microenvironment. This places oligodendrocyte precursor cells in the tumor microenvironment really for the first time, and also asks, I think, very interesting basic questions about what role Neuroligan 3 may play in normal myelin physiology. So the next set of questions that we asked focused on the relative importance of this mechanism. There are many cell intrinsic as well as multiple microenvironmental mechanisms that drive glioma growth and progression. So to test the relative contribution of Neuroligan 3 to glioma progression, we xenografted patient-derived glioma cells into the environment of either the Neuroligan 3 wild type or Neuroligan 3 knockout brain. And what we found was really unexpected. We discovered that these patient-derived xenograft models simply failed to progress in brains that lacked microenvironmental Neuroligan 3. In comparison to the growth that we see in their wild type counterparts. Now, this dependency on Neuroligan 3 extended across multiple different forms of high-grade glioma, from pediatric glioblastoma to diffuse intrinsic pontine glioma and adult glioblastoma, but did not extend to a patient-derived model of breast cancer brain metastasis, suggesting that while this is a really important mechanism mediating high-grade glioma growth, it does not necessarily extend to all brain cancers. So we next turn to that NF1-associated optic pathway glioma model and hypothesized that Neuroligan 3 may also be an important activity-regulated mechanism there. And one thing that really suggested that Neuroligan 3 may be central to NF1-associated optic pathway glioma growth was the observation that in the context of an NF1 heterozygous mouse, as well as a number of other experimental model systems, that there is upregulated Neuroligan 3 secretion in the NF1 mutant optic nerve and retina compared to their wild type controls. So we tested the idea that Neuroligan 3 was a key mechanism here as well, simply by crossing the Neuroligan 3 knockout mouse to this NF1 mutant optic pathway glioma mouse model. And what we found was that just like the effects of dark rearing, that there were many fewer and far smaller tumors that formed in the absence of Neuroligan 3 in the optic nerve, even when the mice were raised with normal visual experience compared to their Neuroligan 3 wild type littermate controls. And so I just told you that Neuroligan 3 is this really interesting therapeutic target for pediatric gliomas, and also that ADAM10 is the enzyme that mediates the cleavage and release of Neuroligan 3 into the tumor microenvironment. And so we tested the idea that by inhibiting ADAM10, we may phenocopy the loss of Neuroligan 3. And excitingly, we found in multiple different patient-derived as well as genetically engineered mouse models that ADAM10 inhibition very starkly slows glioma growth and progression. And so I'm pleased to report that this is a clinical strategy that we've just recently brought to an early phase clinical trial for children with high-grade gliomas throughout the United States. But why is Neuroligan 3 such an important mechanism? You know, what happens in the glioma cell when Neuroligan 3 binds? Well, we find that when Neuroligan 3 is shed in an activity-dependent manner, it binds to a binding partner on the glioma cell we're working very hard right now to identify, and then subsequently recruits numerous oncogenic signaling pathways. There's early and upstream stimulation of focal adhesion kinase, and then downstream SRC, RAS, and PI3 kinase mTOR pathway activation. And this helps to explain the role of Neuroligan 3 as a mitogen. It helps to explain the sufficiency in promoting glioma growth, but it really does not explain this unexpected dependency. And so digging deeper to look at the gene expression changes attributable to Neuroligan 3 binding, we found that Neuroligan 3 regulates glioma cell gene expression and more specifically upregulates a number of different synapse-associated genes. There is a feed-forward effect of Neuroligan 3 on its own expression, together with upregulation of the BDNF receptor TrkB, but also upregulation of a number of different glutamate receptor subunits and other synapse-associated structural proteins. Looking at these gene expression changes in a different way, looking at primary biopsy samples from the major classes of high-grade glioma in collaboration with Mario Suva and Mariella Philbin, we find that in H3K27M mutant diffuse midline gliomas, in IDH wild type, and in IDH mutant glioblastoma, that at the single cell transcriptomic levels, the malignant cells do indeed express very high levels of various forms of glutamate receptor subunits, particularly the AMPA receptor subtype of glutamate receptors, other synapse-associated structural proteins. And now looking at that single cell transcriptomic data in a different way, within each patient tumor, there is a subpopulation of cells that are enriched for synapse-related genes. And when we look at that in a developmental hierarchy plot, it's not surprising to find that this enrichment for synapse-associated genes co-localizes with the more oligodendrocyte precursor cell-like population. But are these, synapses, really happening in both human as well as mouse model tissue? When we look by immunoelectron microscopy to unambiguously identify the glioma cell processes, we see these very clear synaptic structures with a nice postsynaptic density here on this immunolabeled glioma cell process, and then presynaptic neurons across a synaptic cleft containing synaptic vesicles. Very clear synaptic structures, both in primary biopsy samples as well as in patient-derived xenograft models. Looking at these structural synapses a different way and testing the idea that Neuroligan 3 is functioning perhaps in part to promote this neuron to glioma synaptogenesis, we do find that in the absence of microenvironmental Neuroligan 3 that many fewer structural synapses form between neurons and malignant glioma cells. But are these neuron to glioma synapses functional, or are these simply a shadow of the cell types from which these cancers emerge? To test this, we collaborated with Rob Malenka's lab and performed whole cell patch clamp electrophysiology of the glioma cells xenografted into a very tractable, experimentally tractable circuit, the hippocampus. And so this is what the xenografts look like. We xenograft and then about 2 months later make acute hippocampal slices. And then we whole cell patch clamp these GFP positive tumor cells. At the same time, we can stimulate the axonal afferents into that region while we're recording from the glioma cells to see if there are electrical currents in the glioma cells attributable to activity within these axons. And then after we're done making these recordings, we can dye fill the glioma cells that we had patched onto with something like biocytin to go back and just confirm that the cell that we were recording from was indeed a cancer cell and not accidentally, for example, a neuron. And what we find when we do this experiment is that there are indeed excitatory postsynaptic currents in about 5 to 10% of the glioma cells in any given tumor. These are strictly activity dependent. They're blocked by tetrodotoxin. They exhibit the morphology and the kinetics of true excitatory postsynaptic currents, as well as multiple other electrophysiological characteristics of true synapses, including paired pulse facilitation and single vesicle events called mini EPSCs. More specifically, these neuron to glioma synapses are AMPA receptor dependent, blocked by various AMPA receptor blockers. Now, in addition to these true synaptic currents in the glioma cells, we found in every patient tumor we examined another subpopulation of cells that exhibit a different kind of electrophysiological profile. Rather than activity-evoked synaptic currents, we instead saw these activity-evoked prolonged currents that were, instead of being on the millisecond timescale, actually on the second timescale. These were strictly dependent on neuronal activity and blocked by tetrodotoxin. And interestingly, these prolonged currents scaled with the field potential, meaning that the more neurons were active, the larger and longer these currents were. What we determined these currents represent are potassium-evoked currents. They can be stimulated by extracellular potassium, blocked by barium, which blocks calcium channels. Now, we noticed that when we dye filled these cells that exhibit these prolonged potassium-evoked currents, that not just one cell filled, but rather this biocytin dye diffused to a network of tumor cells. And that evoked for us the recent demonstration by Frank Winkler's group in Heidelberg that glioma cells can connect to each other through gap junctions and form kind of this gap junction coupled network. And we wondered whether this gap junctional coupling was actually electrically amplifying these potassium currents. And so we block gap junctions in multiple different ways. I'm showing you the data from meclofenamate blockade of gap junctions. And we find that indeed blocking gap junctions decreases the amplitude of these potassium evoked currents. Now, gliomas have just shown us that there are at least two different ways with which they undergo membrane depolarization. And so I hypothesize that the membrane depolarization alone may be really important for glioma growth and progression. And actually this would make sense because we know that during prenatal brain development, that neural precursor and stem cells depend upon voltage-dependent changes, voltage-dependent mechanisms that are triggered by membrane depolarization changes to regulate proliferation and differentiation. And brain development is aberrant when there are ion channel mutations that modify these voltage-dependent mechanisms. And so we again turn to optogenetics to test this hypothesis that simply depolarizing the membrane of these tumor cells may regulate their proliferation and growth. And what we did is we expressed channel rhodopsin 2 now not in the microenvironmental neurons, but in the cancer cells themselves. And then we find if we shine blue light on these channel rhodopsin 2 expressing pediatric glioma cells, that we can evoke membrane depolarization. And when we xenograph these tumor cells into the mouse brain, we can optogenetically depolarize the tumor. And what we find when we do that is that indeed membrane depolarization increases the proliferation rate of pediatric high-grade glioma xenograph models. Conversely, if we block synaptic signaling, either pharmacologically or in this case genetically by expressing a dominant negative version of the AMPA receptor subunit GluA2, there is a stark effect on glioma growth in vivo. So these tumors are integrating into the neural circuits that they invade. They are doing this both through electrophysiologically functional neuron to glioma cell synapses, as well as electrically through activity-dependent potassium-evoked currents that are then amplified within a gap junction coupled network of glioma cells. We can visualize this electrical activity using genetically encoded calcium indicators that are expressed in the malignant glioma cells. And here you see that when we stimulate the Schaefer collateral afferents into the CA1 region, the glioma cells in that region exhibit these calcium transients indicative of membrane depolarization and inward currents. These depend upon neuronal activity. They're blocked by tetrodotoxin. And of course, there's spontaneous activity within these tumors. And I think this particular calcium imaging movie really underscores the extent to which these cancers are electrically active tissues. And that is not the way we have been thinking about pediatric gliomas. We really need to understand the neuroscience of childhood brain cancers. We have to understand the mechanisms of this malignant circuit assembly, of its plasticity, and of its evolution over the course of the disease. We need to understand what these voltage-sensitive mechanisms that drive proliferation really are. And in doing so, we are going to glean insights for normal neural development and plasticity in the childhood brain through this magnified lens of neural cancers. I want to end the talk with the idea that the communication between neurons and glioma cells is not unidirectional. This is a bidirectional conversation. We've known for a number of years now through Harold Sontheimer's group that adult glioblastoma increases the excitability of neurons in the tumor microenvironment, and this contributes to glioma-associated seizures. More recently, Ben Dineen's group at Baylor has shown very elegantly that synaptogenic factors secreted by the glioma cells really function to promote this cortical hyperexcitability in adult glioblastoma. We found that the same neuronal hyperexcitability exists in diffuse intrinsic pontine glioma. And so this cycle is being set up, a cycle by which the glioma cells increase neuronal activity. The activity of neurons drives glioma growth both through paracrine secreted factors as well as through direct electrical communication. And I think that the mechanistic parallels evident between normal neuron glial and malignant neuron glioma interactions really underscores the extent to which this tumor is simply hijacking mechanisms of normal neural development and plasticity, and demands that we begin to approach these cancers from a neuroscience perspective. In the last moment here, I want to just suggest that the neuroscience of cancer is important not just for central nervous system tumors, but really quite broadly in pediatric oncology. We know that innervation of a variety of different tissues in the body is critical for organ development, for tissue homeostasis and regeneration. And so it's not surprising that there's a number of different groups now finding tissue innervation as a critical modulator of various forms of cancer. Thinking about glandular tissues in adult cancers for a moment, we know that innervation of normal glandular tissue, perhaps best shown in the salivary gland by Sarah Knox and her group, is required for adequate organogenesis. This parasympathetic innervation, in this case, innervates the stem cell niche and drives development of the organ. It's not surprising then that innervation of glandular cancer seems to be critically important. And this has now been shown very, very clearly in a range of different adult cancers: prostate, pancreas, stomach, colon, as well as skin. And I suggest that this is a mechanism likely to be really important across a range of different pediatric cancer types. It simply hasn't been studied yet. But this idea that the neuroscience of cancer is an important pillar that we need to be examining really is underscored by a range of different studies and at a range of levels. You know, we need to be looking for these electrochemical neural cancer interactions, not just in the central nervous system, but also peripherally. Paracrine signaling between neurons and cancer cells, systemic interactions between the nervous system, tumors, as well as the immune system. And then, of course, not to forget the interactions of our cancer therapies on the nervous system and how this plays in, perhaps, actually to tumor progression. There are many people to thank. My laboratory members, past and present, are critically important collaborators of the work that I just showed you, especially David Gutman and Rob Malenka, Mario Suva, and Mariella Philbin, as well as our funding sources. Last but not least, we're very grateful to the patients and families whose donation of tumor tissue enables all of the work that we do. Thank you so much for your time and attention.
Thank you so much, Michelle, for this fantastic talk. It was really in the heart of the topic of this session, and it shows us how it is important to understand the basic mechanism of development. So thank you very much, Michelle. It is the end of this session, and I would like also to thank all the speakers of this session. I will switch to French, sorry, Michel, just to say merci beaucoup à tous. Je voulais juste simplement préciser que c'était la fin de cette session. Et ne pas oublier qu'à la fin de chaque session, nous avons cette discussion qui permet à tous de faire le point et de résumer un petit peu ce qui a été dit au cours des sessions. Donc surtout, n'hésitez pas à intervenir, à poser vos questions. On les relayera et on essaiera d'y répondre. Voilà, donc c'est la fin de cette première journée. Je vous remercie et on se retrouve, si je ne dis pas de grosses bêtises, demain à 10h30. Voilà, au revoir.